So far in this series, we've looked at the genetic component of alcoholism, how alcohol affects the brain, and what motivates alcoholics to continue to drink. In this video, we're going to look at how the brain fights back against the effects of alcohol and what happens to our brain as a result of chronic alcohol abuse. As people begin to chronically abuse alcohol, they start to experience changes in their brain chemistries. Depending upon how much and how often a person drinks, the constant actions of alcohol on the brain will impair the brain's ability to respond and function normally. Over time, the effects of alcohol and the resulting changes in brain chemistry can cause a wide range of unpleasant experiences, ranging from temporary effects to permanent damage. In response to alcohol's negative effects on its functions, the brain protects itself by taking a series of countermeasures so that it can continue to operate as normally as possible. Ironically, while these countermeasures are intended to protect us, for chronic alcoholics, they can actually make abstinence more difficult. To find out more, let's take a closer look at how the brain adapts to the effects of alcohol, beginning with a process called homeostasis. One of the most amazing aspects of the human body is its ability to adapt to changes in the environment that may threaten its normal function. The brain is no exception. Alcohol is a mood-altering drug that works by interfering with the normal functioning of the brain. Sensing this interference, the brain attempts to counteract the effects of alcohol so it can continue to function as normally as possible. The technical term for this process is homeostasis. To understand how homeostasis works, let's quickly recap how alcohol impairs the brain. As we've previously seen, the human brain is made up of many millions of cells called neurons, which, when functioning normally, use a combination of electrical signals and chemical messages to form a complex communications network. The wall of the neuron is made up of two layers of lipid molecules called the membrane bilayer. The membrane bilayer provides support for ion channels that control the ratio of positive and negative ions within the neuron. Neurons need a carefully controlled balance of these positive and negative ions to function normally. When we drink alcohol, the alcohol molecules quickly reach the brain and penetrate the membrane bilayers of neurons. Alcohol molecules insert themselves between the lipid molecules that form the membrane bilayer. This causes the ion channels to leak more ions into the neuron, disrupting the correct balance of ions in these cells. When a neuron no longer has a controlled balance of positively and negatively charged ions, it ceases to function properly, resulting in a condition of mental and physical impairment. This process of impairment begins as soon as alcohol molecules begin to enter the membrane bilayer, making it less stable and more fluid. Normally, the membrane bilayer is fairly stable. The lipids that make up much of the membrane are tightly packed and lay locked in place head to head and tail to tail. Fluidization occurs because the alcohol molecules are much smaller than the lipid molecules. When alcohol molecules insert themselves into the membrane bilayer, they create more room for the lipid molecules to freely move their tails, resulting in a membrane that's more fluid and less stable. The brain responds by activating a homeostasis mechanism in the form of a chemical reaction whose mission is to repair the damage done by the alcohol molecules. For successful adaptation to occur, the neurons must counteract fluidization and work to stabilize their membranes so that they can continue to function as normally as possible. There are several ways in which the neurons can fight back against the alcohol. Most of these ways involve changing the construction of the membrane as a sort of antidote against the invading alcohol molecules. The lipid molecules in the membrane can have several different types of tails. Some are made of saturated fatty acids, while others are made of unsaturated fatty acids. The unsaturated tails are much stiffer. Therefore, if a membrane contains more lipid molecules with unsaturated tails, it will be more rigid than membranes with more saturated tails. Usually, the neurons will first try to stiffen their membranes by increasing the number of unsaturated lipid molecules. Because the unsaturated lipids are stiffer, they help restore stability to the membrane. But when higher and higher amounts of alcohol are present, the neurons are forced to take more drastic steps. Now, the neurons work to stiffen their membranes by inserting cholesterol into the bilayer. Because of their size and stiffness, these large honeycomb-shaped molecules are able to help reinforce the membrane and restore its stability. Whether it's increasing the proportion of lipid molecules with stiffer tails or inserting cholesterol molecules, the brain uses a number of homeostasis mechanisms to counteract fluidization and keep the membrane bilayer stable. Most of the time, these efforts are successful, but not always. Chronic heavy drinking makes homeostasis more difficult to achieve and can force the brains of people who abuse alcohol to take actions that become more and more extreme and more dangerous. As we've seen, homeostasis is an important weapon by which the body adapts to the effects of alcohol and reduces its effects. But as we're about to see, because it is so effective, homeostasis can actually make it more difficult for an alcoholic to achieve abstinence. If homeostasis is so effective at shielding the brain from alcohol's effects, how can it possibly get in the way of abstinence? Well, the answer is indirectly. And here's how. Homeostasis is what leads to two important and very negative effects of alcohol: tolerance and withdrawal. Together, these two effects can unfortunately make it easier to drink more alcohol, while at the same time making it difficult to drink less. Homeostasis provides the biological basis for how alcoholics develop tolerance to alcohol when they continue to drink, and why they experience withdrawal symptoms when they quit drinking. The most common definition of tolerance is when a person has to drink more and more alcohol to reach the same level of intoxication. In other words, a person who is tolerant to alcohol gets less of an effect from the same amount of alcohol than someone who is not tolerant. Drinkers develop tolerance because homeostasis works to lessen the impact of alcohol on the brain. For example, as homeostasis makes the nerve cell membrane stiffer and stiffer, it takes more and more alcohol to fluidize the membranes, disrupt GABA channels, and release more dopamine in the reward pathways. Over time, then, drinking the same amount of alcohol produces less and less of a reinforcing effect. So to get the strong reinforcing effect they are used to, individuals who are tolerant have to keep drinking more and more alcohol. This begins a dangerous cycle of more drinking causing more tolerance, which leads to more drinking, which causes even more tolerance, which leads to, well, we know where this cycle leads, and it's not good. This cycle eventually causes the brain to lose its sense for where its chemical equilibrium should be. At this point, the brain can actually require alcohol to function normally. This also explains why many alcoholics feel like they have to drink to feel normal. Because homeostasis has by now severely stiffened the membrane, it may have become so rigid that it actually needs alcohol to fluidize it back into a normal range of functioning. At this stage, the brain has not only adapted to the chronic presence of alcohol, it has become physically dependent on the presence of alcohol to function normally. This dependency is so strong that a sudden withdrawal from alcohol will cause the brain to malfunction. Once the brain has adapted to the depressant effects of alcohol, when alcohol is withdrawn, the brain becomes overactive and its neurons are now much more likely to fire. The technical term for this overactivity is a rebound effect. The rebound effects that occur in alcoholics who have stopped drinking range from uncomfortable to dangerous. Many rebound effects can be linked to specific areas of the brain with symptoms that are the opposite of alcohol's pharmacological effects on these brain areas. For example, the cerebellum controls motor coordination. When affected by alcohol, it causes a loss of balance and coordination. The effects of alcohol withdrawal on the cerebellum include muscle tremors and sometimes seizures. The cerebral cortex controls judgment and decision-making. Alcohol's effects on the cortex lead to inappropriate responses to situations. The effects of alcohol withdrawal on the cerebral cortex include confusion, disorientation, and hallucinations. The reticular activating system controls our level of physical and psychological arousal. When affected by alcohol, it causes our reactions to become slower. The effects of alcohol withdrawal on the reticular activating system include excessive arousal and agitation during the day and sleeplessness at night. The limbic system controls our emotions and helps to determine our appetite for rewarding activities such as eating or sex. The effects of alcohol withdrawal on the limbic system include mood swings and cravings for alcohol. Once an alcoholic stops drinking, the onset of withdrawal symptoms usually begin to occur within 8 to 12 hours. Initial symptoms include nausea, headache, heart palpitations, and tremor. Over the next few days, a person in alcohol withdrawal may experience more severe symptoms. These may include seizures, hallucinations, and delirium tremens, which is a potentially life-threatening condition caused by severe instability in the nervous system. The dangers from the most severe withdrawal symptoms are typically over within a week or two. However, it may still take several weeks for an individual to start thinking clearly again and for his or her depression to lift. Other symptoms such as cravings, mood swings, insomnia, and irritability may linger for months. Fortunately, given both time and abstinence, most of the changes in brain chemistry caused by alcohol can be reversed. With abstinence, the brain's neurons regain their normal balance of saturated to unsaturated lipids in the membrane. Also during this time, the cholesterol that was used to stiffen the membrane will be removed. However, for some alcoholics, some effects of alcohol on the brain can unfortunately be permanent. For example, the cerebral cortex, the brain center that controls judgment and decision-making, can permanently shrink once it has been damaged by alcohol. This damage to the cerebral cortex can result in alcoholic dementia, an impairment of thinking, learning, and memory. Although alcoholic dementia is not very common, for those individuals who do experience this condition, it can be devastating. Once they abstain from alcohol, a small number of alcoholics with dementia may experience some reversal of their impairment, but for most, the damage is permanent. Another rare form of alcohol-related brain damage is Wernicke-Korsakov syndrome, or WKS for short. Also known as alcoholic amnestic disorder, WKS causes severe memory loss and disorientation. Alcoholics afflicted with WKS sometimes lose their ability to think abstractly, making abstinence more difficult because they are unable to link drinking to their symptoms. Unlike alcoholic dementia, Wernicke-Korsakov syndrome is not caused by the direct actions of alcohol on the brain. Instead, WKS is caused by a lack of thiamine, which is also known as vitamin B1. This and other vitamin deficiencies result from both the poor nutritional habits of many alcoholics and the reduced absorption of nutrients that occurs as a result of chronic alcohol drinking. While many alcoholics with Wernicke-Korsakov syndrome will improve once they abstain from alcohol, about half will not. The cerebellum, which is responsible for coordination and balance, can also be damaged by chronic alcohol drinking. The cause of alcoholic cerebellar degeneration is thought to be a combination of the toxic effects of alcohol and a thiamine deficiency. Although abstinence will prevent worsening of the condition, any existing damage is permanent. We've learned that chronic alcohol drinking and the brain's use of homeostasis to protect itself from alcohol is what leads to the development of tolerance, rebound effects, and withdrawal symptoms. We've also discovered how the brain adapts to chronic alcohol abuse by changing its chemistry, and that sometimes the adaptation is so severe the brain will actually require alcohol to function normally. Lastly, we've seen that alcohol has several acute and chronic effects. The acute effects are caused by fluidization of the membrane and interference with the functioning of important ion channels in the membranes. The good news is that, given both abstinence and time, most of the changes in brain chemistry caused by chronic alcohol abuse can be reversed in a matter of weeks. The bad news is that some chronic alcoholics can suffer permanent impairments to their thinking, memory, and coordination. Because of the brain's unique ability to adapt its chemistry, it is capable of successfully responding to the effects of alcohol through a process called homeostasis. However, just as with homeostasis mechanisms in other systems of the human body, there are limits to the brain's ability to respond to chronic, heavy drinking. By exceeding the body's ability to respond effectively, alcohol abusers become at risk for a variety of neurological and other medical problems.